right. So today at our house, Pechacucha, we're delighted to welcome today Pere Gelabert. And Pere is originally from Spain, and many of you know him already, so I won't give a long-winded introduction. But he did his undergraduate and master's and PhD in Barcelona at two different universities in biology, biological anthropology, bioinformatics, and biomedicine. And then he came here to Vienna as a postdoctoral researcher, initially working with Ron Pinhazi as a postdoc in the Department of Evolutionary Anthropology, where he's now staying and based. From 2023 on, he was an FWF principal investigator with a exciting FWF grant. And then he succeeded in getting an ERC starter grant this past round, but one, I think. And now he's a tenure track a researcher here in the department in the University of Vienna. And so Pere's doing some really exciting cutting edge research in ancient DNA and things like sediments and bones and so on and other things. And what's really exciting today, which I love, is that he's going to give a talk in which it's I'm going to be touching upon some of the seed grant work that he was doing with Manasij Pal Chowdhury with paleoproteins and sediment DNA. So without much further ado, let's hand it over to Pere. We're looking forward to your talk, and we'll have some questions at the end for anyone who's been paying attention. You can share.
Okay. Thank you very much for being here. Today I'm going to present the early populations of Patagonia, a global approach. This is a very big project that has been ongoing for the last 3 to 4 years, which has received has seed funding, which I am super grateful on a part of the project. And today I'm going to try to present the whole picture of the project. The project happens in Patagonia, which is the southernmost part of America. Patagonia was settled as any other location in America through migration corridors. In the case of Patagonia, most of the human waves that settled Patagonia came from the Andes, so from the Pacific coast of Patagonia. And Patagonia is divided into two regions, north and south. The oldest occupations in the northern part of Patagonia are dated to 14,000 years ago, and the oldest occupations in the southern part of Patagonia are dated 13,000 years ago. Generally, it has been understood that the cultural transitions of this region are associated with population continuity, and also there is very limited amount of archaeological sites. Most of the research has been focused on quite historical populations, such as the famous Yamana or Cauesa, which lived in Tierra del Fuego, in southernmost part of Chile and Argentina. And this is basically the research that has been used to demonstrate that most of these transitions will be with population continuity. That is, in fact, not against to the evidence that we already have from at least 2 separate individuals, one from Chile and one from Peru, that several waves of human colonization in the early Holocene or late Pleistocene in America did not result into inheritance of further generations. That means that we have already identified at least 2 or 3 dead-end populations. So this is what we wanted to assess. In order to assess these initial waves of colonization in Patagonia, we studied Baño Nuevo 1 site, which is a very important and famous site in northern part of Chile, and Aysén, that we visited several times. These are pictures from 2023, when Olivia and myself, we went there for sampling humans and sediments. The site is located in an ancient glacier in the region of Aysén, and it's very important for 2 different things. First, because it's one of the few caves that exist in northern Patagonia. There are very few caves. And second, because it's one of the sites that has the oldest dates in northern Patagonia, but also continuous occupation, which makes it a perfect site to study not only the earlier waves of human occupation in Patagonia, but also transitions. Here on the left, you can see an image where the cave is located. In red, you can see the entrance of the cave. And on the right, you can see how the cave looks like. The cave is quite a pretty little cave. It has been mostly excavated, except for the entrance, where there is the archaeological profile that we studied. Also, it's remarkable to mention that this cave has 10 human inhumations identified so far, which converts it to the oldest collective burial, not in Chile, but in the whole American continent. All these 10 graves come from stratigraphic unit 3, dated to 10 to 11,000 years ago, which is above stratigraphic unit 4, 5, and 6. Stratigraphic unit 4 is human, 5 is paleontological. All these individuals have been radiocarbon dated, all having coincident dates around 10,200 years old, which basically hints towards the existence of a homogeneous group that lived there 10,000 years ago. So here we can do questions about kin organization, genetic ancestry, and also effect on fauna. We try to sequence these 10 individuals. We succeed to get genomic material from 6, which we could recover the mitochondrial haplogroups, already evidencing diversity within the diversity known in the Americas. All these individuals show also typical deamination patterns, but quite good preservation of DNA. That good preservation of DNA that we could recover genomic material from 3 individuals that already doing the calculations that we did now, we are sure that we can bring 3 individuals to high coverage. That would be a huge achievement, because if that will succeed, that would be the oldest genomic data at high coverage from South America. And also, it would be very important to address all these questions that we want to address. When we have studied the genomic material that we have now from these 3 individuals, it's clear that they not belong to the same kin, so they're not related at first or second degree, and they seem to cluster with the oldest genomes from South America, but not from the Patagonian genomes, meaning that most likely these individuals represent one of these populations that did not succeed, and it's a dead end, genomically speaking. This is why we need high coverage data, because one of the key questions is what is these people representing? Which population do they represent? And for doing that, we need high coverage data. We need to do demographic modeling. We need to understand the real diversity of these individuals in context with the rest of information. This is why we will continue working on this. Apart from that, to have a much more comprehensive idea of this site, we also got sediment DNA. So we got 70 sediment samples from the archaeological profile, very far away from the burials, from square 7V, in order to avoid any kind of turbulence or disturbance of the profile. The samples cover the whole stratigraphic profile, and we're interested in basically identify when humans arrived in Baño Nuevo, if it happens in stratigraphic unit 4, which there is archaeological material, or in 5, which is theoretically paleontological, which humans are present in stratigraphic units 4 and 2, so that's and beneath and above the burials. And do we see any relationship between fauna and humans? So far, through what we call a low-cost method, we have identified very good DNA preservation in the site, at least in the samples that we have identified. We don't have final results because this is a super laborious work, and I didn't have anyone to do that. So this is going to be the future work of one of my PhD students, Pere Gelabert, who I'm sure that in the future she will provide you with plenty of very exciting results of Baño Nuevo. And just to bring another piece of complexity to this whole portrait, we are also committed to doing zooms analysis, which would be one of the first applications of zooms in South America, to provide a way more detailed representation of the archaeofauna, especially focusing on extinct mammals, but also on the relationship between humans and fauna through ctDNA and zooms. This is happening in 2 different places. The samples are being prepared by Francisca Santana at the Catholic University of Chile, where the collagen is extracted and the samples prepared, and then Manajit here at the proteomics lab at the University of Vienna. It's going to analyze these samples. We hope that really soon in time, and we are going to be able to compare that with the archaeofaunal representation and also with the ctDNA analysis. Just with that, I would like to thank everybody from the collaborating institutions, from the Pontificia Universidad Católica de Chile, Francisca, for the people in the research center in Aysén, César Méndez and Amalia Nuevo, for the rest of people of the department that has been helpful, and of course to the people of my team that are deeply committed to this project and they are working on it. Thank you very much for your attention.